Hello, my name is Cindy Atwell, and I'm VP of Business Development at Precision BioSciences. We are a clinical stage biotechnology company, leveraging a next-generation genome editing platform for the development of therapeutics. We are a public company, and I will be making forward-looking statements today. Please refer to the disclosures and risk factors in our SEC filings. Precision BioSciences is dedicated to improving life through the application of our proprietary Arcus genome editing platform. The focus of Precision BioSciences is to create therapeutics that address unmet medical needs. Applying Arcus, we are seeking to overcome cancer through the development of allogeneic CAR T therapies and to cure rare diseases through in vivo gene correction. Our wholly owned subsidiary, LO Life Systems, also applies the Arcus platform to improve human health through food. It has been an exciting year at Precision, during which we have built a world-class team of 220 employees. We've established a GMP manufacturing facility and, importantly, have smoothly transitioned from a preclinical to a clinical development stage company with now 3 allogeneic CAR T therapies currently in clinical studies. Our proprietary genome editing platform, Arcus, is the underlying foundation of Precision BioSciences. So what is Arcus? Arcus was invented at Precision and is the only gene editing technology developed with a focus on clinical translation of therapeutics without unintended changes to the genome. Our genome editing pioneers looked to nature for a better editing system, better specificity, accuracy, and versatility. Our team started with a natural enzyme called ICRE1, which evolved in nature specifically to perform precise gene edits in a eukaryotic organism, algae. We developed a process to engineer ICRE1 and reprogram the enzyme to target new genetic sites. This is what we call Arcus. Arcus has 4 key advantages. Safety. The enzyme has a natural off switch and doesn't edit DNA unless it's productively bound to its target site. Delivery. Arcus is only 364 amino acids long, and the small size permits delivery either by LNP or AAV. Control of the type of edit. Because of the unique type of cut, Arcus can be applied to efficiently knock in or knock out target genes of interest. And it is proprietary. Precision has full control and freedom to operate. In July 2019, we opened MCAT, a fully GMP compliant facility. We chose to invest early in manufacturing, believing this to be a strategic advantage. We're able to produce all key components across the portfolio for CAR T and in vivo gene correction therapeutics. And we are currently producing clinical trial material for our anti-BCMA CAR T program. I'll now provide an overview of our 2 therapeutic business areas, starting with our allogeneic or off-the-shelf CAR T therapies. Cell therapy for cancer is one of the major breakthroughs of modern medicine. There's been impressive efficacy in several blood cancers and in treating patients where other options have failed. However, several key limitations remain, including accessibility, cost, logistics, variability, and safety remains a concern. We believe there is a compelling opportunity to use gene editing to improve on this, which is the core focus of Precision today. Allogeneic CAR T is still early in development as a therapy class, but between our work and others, early evidence of encouraging clinical activity has emerged. Coupled with this is the opportunity to expand the reach and patient access of cell therapy. And we can start to think about this more like a traditional drug that could ultimately be used in a community oncology setting. So what is needed for a successful allogeneic CAR T product? There are 4 key elements. 1, manufacturing. It's critical to be able to produce consistent cell product at scale and a cost of goods more like a biologic. 2, CAR T cell phenotype. There's growing evidence that the type of T cells in the final product matter for overall clinical activity. 3, a safety profile that supports an off-the-shelf product. And 4, clinical activity. The overall profile needs to be effective in killing cancer. So how is Precision measuring up to these 4 key attributes? 1, manufacturing. We're able to consistently manufacture high-quality GMP CAR T cells at scale. Secondly, our drug product consistently comprises at least 50% naive and central memory T cell phenotypes. Third, as we reported last year, while early, we've seen a very encouraging safety profile with no graft versus host disease, no severe CRS or neurotoxicity, and importantly, no infections due to lymphodepletion. And lastly, we saw encouraging early evidence of clinical activity, even at very low dose levels. It has been a big year for Precision. We transitioned from a preclinical into a clinical development stage company. In just about a year and a half, we have launched 3 clinical trials. And part of the reason that we were able to do so is that there is a lot of similarity across the 3 therapeutics that we have put into the clinic. Namely, the Arcus enzyme we use is the same and the manufacturing process is similar. We simply switched out the gene inserted, which is the CAR targeting the antigen relevant to the type of tumor we're aiming to treat. This allows Precision to rapidly apply its learnings across our cell therapy platform, as well as to rapidly develop new off-the-shelf CAR T therapies. Last December, we were excited to present early data from the PBCAR-0191 trial from 9 patients treated at the first 2 dose levels. To remind everyone, the safety profile we observed at dose level 1 and dose level 2 is very encouraging. There were no dose-limiting toxicities seen, no graft versus host disease, and no grade 3 or higher CRS or neurotoxicity. And again, importantly, no infections from lymphodepletion. Turning to clinical activity, it's important to consider this patient population generally had a high tumor burden and aggressive disease. Recognizing that this is an early look from a phase 1 trial and a small number of patients at very low doses, these data supported dose-dependent clinical activity with PBCAR-0191. We have illustrated some key results in the non-Hodgkin's lymphoma cohort on this slide. Across dose level 1 and dose level 2, we saw 4 out of 6 patients respond at day 28 or beyond, for an overall response rate of 67%. With this encouraging early safety and high response rate, we are applying 3 levers to drive towards durable responses. The first is increasing dose to drive peak expansion. The second, we are testing novel lymphodepletion regimens to extend the window of allogeneic cell persistence. And third, we are repeat dosing to eliminate remaining tumor cells. Essentially, we are taking a pharmacological approach to cell therapy with the goal of more closely approximating the durability of autologous CAR T while maintaining a predictable safety profile that could enable uptake in multiple lines of therapy and various treatment centers. To summarize the off-the-shelf cell therapy area of the business, we think we can achieve an allogeneic cell therapy which delivers the favorable safety and accessibility profile of a traditional biologic with that CAR T efficacy. We think this will be extremely compelling for patients and physicians, and we hope with the potential to drive widespread uptake in multiple centers and lines of therapy. We have focused the discussion today on blood cancers, but we also see tremendous potential in applying Precision's allogeneic cell therapy platform for the treatment of solid tumors. Turning now to our in vivo gene correction portfolio, we are using Arcus to create potential cures for various genetic and infectious diseases. We believe there are 4 core elements that are required for real success when deploying gene editing directly into the body of a patient to treat disease. The first and perhaps most important is safety. It is critically important to minimize the occurrence of off-target editing, which can drive unintended genetic changes and adverse events. Secondly, we need to be able to effectively deliver the editing technology to the cells and organs of interest. And third, the editing must be efficient at the target site of interest. And lastly, the editing that is achieved must drive a potent therapeutic effect in the patient. And it is here where we believe large animal data is critical to understanding the true clinical translation that can be achieved with an editing technology. So how does our Arcus platform stack up against these 4 criteria? Illustrated on this slide are data relating to our PCSK9 program, which was published in Nature Biotechnology in 2018. We believe Arcus is one of the most precise editing technologies in existence today. You can see in panel 1 that the 3rd generation PCSK9 nuclease has 0 detectable off-target editing. Moving to panel 2, Arcus is very easy to deliver, mainly because it is so small and easily fits into either an AAV vector or LNP. Illustrated in panel 3 is the powerful editing efficiency which we have achieved in non-human primates, knocking out the PCSK9 gene. And as you can see in panel 4, this is highly therapeutically relevant, driving more than a 90% reduction in PCSK9 in these animals. We believe this effect to be permanent and inherited by the daughter cells. In the PCSK9 non-human primate study, we have demonstrated the durability of the effect for over 3 years and counting. Finally, just to remind you, the Arcus platform is entirely proprietary to Precision and protected by more than 50 patents worldwide. Our liver-directed in vivo pipeline is illustrated on this slide. Our most advanced program is using Arcus to develop a potential cure for chronic hepatitis B infection. We had a productive collaboration with Gilead on this program, which concluded earlier this year. We enjoyed an aligned relationship and are grateful for the opportunity to advance our Arcus technology as well as our potential cure for HBV. As we have regained the full rights and data to the program, we are actively exploring new partnership opportunities for this asset. Earlier this year, we also announced that we will be focusing on a disease called primary hyperoxaluria type 1, or PH1, as our lead wholly owned in vivo program. Over the course of the next slide, I'll tell you a little bit more about this disease and our approach. Primary hyperoxaluria type 1, or PH1, is a rare genetic disease which is driven by a mutation in the liver, which manifests itself predominantly in the kidneys. Patients often present with end-stage renal disease, by which point the only treatment option is a combined liver-kidney transplant. Our approach is to use Arcus to develop a one-time permanent cure for PH1. We decided to move forward with PH1 as Precision's wholly owned lead program based on compelling animal data that we have generated in part through a collaboration with Dr. Wilson's group at the University of Pennsylvania. We are expecting to identify a clinical candidate for this program by the end of the year. In conclusion, we are very excited about what we have already achieved at Precision, as well as what is to come. Our company is made up of a tremendous team, including global pioneers in gene editing who have developed a truly differentiated gene editing platform, Arcus. We have invested early in manufacturing to give us strategic control across the portfolio. Leveraging Arcus, our 3 business areas are progressing rapidly. We have already generated clinical data that validates our approach in allogeneic CAR-T and are advancing a portfolio of in vivo gene correction programs addressing areas of significant unmet need. We are well funded and have cash into 2022, which will allow us to continue to push forward to realize our vision of improving life through genome editing. Thank you for your attention today. Given the time constraints, this presentation included only a subset of the information found in our full corporate slide deck. Please visit the Precision BioSciences Investor Relations webpage for the full corporate slide deck, as well as the most up-to-date information on the company and pipeline.